In this video, we're going to talk about mitosis. So what is mitosis? Well, mitosis is going to be the division of cells. And there are going to be several stages of mitosis. And here in bold, we have the stages of mitosis. And then we're also going to have interphase and cytokinesis. And interphase is going to happen before mitosis. It's our preparation for mitosis. And then cytokinesis is going to happen after mitosis. And we're going to talk about each of these stages in just a second. And then I have a little mnemonic here that I use to remember which stage comes when. So I say, in Paris, my aunt throws cucumbers. So it's very silly, but it helps me remember that interphase, prophase, metaphase, anaphase, telophase, and cytokinesis is the order in which mitosis happens. First, let's take a look at interphase. So interphase is going to be our cell growth and preparation for division. And remember, this is not actually a stage of mitosis. It's just our preparation for mitosis. So we're going to have several stages of interphase. We're going to have G1, and we can see that right here. It's just normal cell growth. Normal cell growth. Next, we're going to have the S phase. And in the S phase, we're going to have DNA replication. And we're going to talk more about DNA replication in another video. So the next we're going to have G2, and this phase is just growth and preparation for mitosis. So we have G1, S, and G2. And we're going to have multiple checkpoints along the way. And at these checkpoints, the cell is going to determine if it's ready to replicate. And this is going to ensure that it does not replicate if the cell is damaged, because we don't want more cells that are damaged, because those damaged cells would then go on to replicate, and we would have even more damaged cells, and that would not be good. So we're going to have a checkpoint at the end of G1, and if we get to the end of G1 and our cell is damaged, it's not ready, then our cell is going to shut it down, and it's going to say, we're not going to divide right now. So if everything is good, we're going to proceed on. Our DNA is going to replicate. Our cell is going to grow. But if we get to the end of G2 and something is wrong, then our cell is going to say, nope, not ready to replicate. We're going to stop right here. If everything is good, then we're going to proceed on. And we're going to have one more checkpoint during metaphase. And this is a stage of mitosis. So if we're in the middle of mitosis at metaphase and something is wrong, then our cell is going to say, nope, not ready, and it's going to stop mitosis. And then here we have a cell that is in interphase, and we can see the nucleus right here. It's all a solid color. Our chromatin has not condensed into chromosomes yet that we can see with our microscope. So it's all just one solid color. So the first stage of mitosis is prophase. This is the first stage of mitosis. And during prophase, our chromatin is going to condense, and that's going to make it easy for the cells to separate the sister chromatids. And we have right here, this is a duplicated chromosome, and each side is a sister chromatid. These are identical. And connecting our sister chromatids is a centromere. And we're going to see that in just a second. So during prophase, the nuclear envelope is going to dissolve. You can see that right here. Our nuclear envelope is starting to dissolve. And centrosomes are going to move to opposite sides of the cell. And centrosomes are made up of two centrioles. Centrioles are organelles in our cells. So we have these centrosomes. They are moving to opposite ends of the cell. And mitotic spindles are coming out of these centrosomes. And in late prophase, we're almost to the next phase, but not quite yet. So in late prophase, the microtubules here, they're going to attach to the centromeres, and that's what's holding together these two sister chromatids. So the microtubules, that's part of the mitotic spindles, they're going to attach to the centromeres. So then we're going to go into metaphase. In metaphase, these sister chromatids, they're going to line up in the middle of the cell. And we can remember that M is for metaphase, M is for middle. So in metaphase, these sister chromatids are all in the middle of the cell. And we can see these microtubules connected here to the centromeres. Then next, we're going to go into anaphase. And in anaphase, these sister chromatids are pulled apart. And I remember that A is for anaphase, A is for apart. So in anaphase, our sister chromatids are going to be pulled apart here. So they were in the middle during metaphase, and now we're in anaphase. They're pulled apart. And we can see these microtubules that are connected to the sister chromatids here. Next, we're going to go into telophase. In telophase, the nuclei, the nuclear envelopes, are going to begin to reappear. We can see that right here. We have two nuclear envelopes that are beginning to appear here. We're also going to have a cleavage furrow that is going to form. You can see that right here. And this is where the two new cells are going to form. Our mitotic spindle is also going to break down. And remember, the mitotic spindle, those were connected to the sister chromatids here. And so that is going to break down. We're no longer going to have that. So this is the last stage of mitosis. And our chromosomes here, they're going to begin to unravel into chromatin again. Finally, we're going to have cytokinesis. And remember, cytokinesis is not actually a stage of mitosis, but it is necessary for the cells to divide. So in cytokinesis, we're going to have the division of the cytoplasm and of the organelles. And the cells are going to separate. And we can see the nuclei right here. And we can see they're represented with a solid color because all of those chromosomes have unraveled into chromatin, and we can no longer see it with our microscopes. So these cells are going to divide, and we're going to have two separate cells now. And that is the end of cytokinesis and the end of our cell division. So now let's look at a summary. So remember, interphase, that was before mitosis happens, and that is cell growth and preparation for division. And we're going to have those checkpoints, and if at any point the cell is not ready to divide, we're going to shut it down, and we're not going to go through with mitosis. But if everything is going well, then we're going to go into mitosis right here, and our first stage of mitosis is prophase. And remember that chromatin is going to condense into chromosomes, and we can see those with our microscope. So when you can see those chromosomes, you know you are in prophase. We've started mitosis. Next, the cell is going to go into metaphase. And remember, the chromosomes are going to line up down the middle. M is for middle, M is for metaphase. So in metaphase, all of these chromosomes are lined up down the middle of the cell. In anaphase, those chromosomes, the sister chromatids, are going to pull apart. A is for apart, A is for anaphase. So in anaphase, those chromosomes, or those sister chromatids, they're going to pull apart. And then our last phase of mitosis is telophase. And in telophase, those chromosomes are going to be at opposite ends of the cell. They're going to begin to unravel into chromatin. And the nuclear envelope is going to begin to reappear. And sometimes it's hard to tell when you're looking at a cell when the cell is going from anaphase to telophase. But if you can see that nuclear envelope beginning to form, then you know you are in telophase. And that's the last phase of mitosis. And then we have cytokinesis. And cytokinesis is going to be the division of the cytoplasm and the organelles. And that's going to be necessary for those cells to divide. And that's going to form two new cells.